70%. I mean, that is just a massive, terrifying proportion of all human cancers.
It really is. And they're all driven by this single hyperactive gene.
Right, the MYC gene, MYC. We have known about this gene for, what, decades now. It is everywhere.
Oh, absolutely everywhere.
And yet, despite throwing billions of dollars and just countless hours of research at it, we have never been able to develop a single drug that targets it directly. It is like the ultimate undruggable mastermind of oncology.
Yeah, and it is incredibly frustrating for researchers. You are looking right at the culprit, but because of the biological neighborhood that culprit lives in, you simply cannot take a direct shot without causing catastrophic collateral damage.
Which is exactly why today's deep dive is going to look at this problem from a completely different angle. We're digging into a stack of recent research, anchored by a really groundbreaking study from Nature Communications.
Yeah, this team of genetic detectives basically decided to stop looking for a direct shot at MYC.
Right. Instead, they built a massive genome-wide in vivo CRISPR screen, basically to find a hidden backdoor, a secret vulnerability in these deadly MYC-driven lymphomas. Okay, let's unpack this, because setting up a trap for a cancer driver you can't actually touch requires some absolutely brilliant biological engineering.
It does. And to appreciate the trap, I think we first need to understand why the mastermind is untouchable to begin with.
Right. Why can't we just drug it?
Well, MYC is a transcription factor. Its job isn't just to cause cancer. I mean, its day job is to act as a fundamental manager for the entire cell.
So it's regulating everyday essential processes.
Exactly. We're talking about basic cell division, how a cell differentiates into its final form, basic metabolism, and even the natural programmed death of a cell.
So if you design a drug that just turns MYC off completely, you wouldn't just be starving the tumor.
Oh, no. You would shut down healthy cellular functions across the entire human body. Yeah, it is just too deeply woven into the fabric of normal life. So the researchers knew direct inhibition was a dead end. They needed to find what we call a synthetic lethality.
Synthetic lethality. So in simpler terms, they needed to figure out what other genes MYC relies on when it goes rogue and starts a tumor.
Right. If you can find the sidekick, you might find a target you can actually drug.
Getting to that sidekick involved a mind-blowing experimental setup. They used something called the MYC transgenic mouse model. Let's break down what that actually means, because it sounds like a serial number.
It does, yeah.
So EMU is a genetic switch, an enhancer, right, that normally drives the production of antibodies in your immune system's B cells.
Spot on. And in this specific laboratory mouse, scientists took that really powerful antibody-producing switch and they wired it directly to the c-MYC gene.
So as these B cells are developing in the mouse's bone marrow, instead of pumping out useful antibodies to fight infections, what happens?
They are tricked into pumping out massive, just unnatural amounts of the MYC protein.
Which creates a massive biological traffic jam. You end up with a huge backlog of these early immature B cells.
Yeah, they are multiplying rapidly because MYC is pushing them to divide, but they can't mature into functional immune cells.
No. Normally, the body has a defense mechanism against this, doesn't it?
It does. When a cell realizes it is multiplying wildly and not maturing, it triggers a process called apoptosis.
Programmed cell suicide.
Right. The cell senses something is deeply wrong and just takes itself out.
But in this traffic jam, eventually, usually around, 140 days into the mouse's life?
Yeah, roughly 140 days.
One single cell out of millions acquires a spontaneous secondary mutation, like a partner in crime that breaks that suicide switch.
And the moment that happens, you get an explosion of monoclonal lymphoma, full-blown cancer.
And that 140-day mark is the baseline.
Right. Now, to find MYC's vulnerabilities, the researchers wanted to systematically mutate genes to see what would happen. But, and this is key, they didn't do this in a standard laboratory petri dish.
They did it in vivo.
Yeah.
Inside living mice. Wait, I have to interrupt here, because doing genetic screening inside a living, breathing mouse sounds insanely expensive, incredibly complex, and really slow. Why wouldn't they just take some of these MYC-driven cancer cells, put them in a plastic dish, and use CRISPR on them like every other lab does?
Because a plastic dish is a terrible flat imitation of a living body.
How so?
Well, a petri dish provides unlimited nutrients and zero threats. It completely lacks a tumor microenvironment.
Right, there's no immune system.
No complex immune system actively hunting the cancer cells. There are no fluctuating blood oxygen levels, no metabolic scarcity.
It's too easy for the cancer.
Way too easy. If you want to find a real clinically relevant tumor suppressor gene, a gene that is actively holding back cancer in a living organism, You have to hunt for it inside a living organism.
So they engineered a biological gladiator arena. And the mechanics of this screen are wild. Let me walk through this. They started by taking primary fetal liver cells from mouse embryos.
Yes, at exactly day E13.5 of development.
Day 13.5. They picked that exact half-day mark because at that stage, the fetal liver is an absolute goldmine of hematopoietic stem in progenitor cells.
HSPCs, yeah. Those are the granddaddy stem cells that eventually create every single blood and immune cell in the body.
And these specific embryos were already engineered to carry the overactive MYC gene, plus the Cas9 protein, right?
Right, Cas9 being the molecular scissors used in CRISPR. So they took those stem cells and introduced a massive viral library containing 87,987 different single guide RNAs.
Over 87,000.
Think of a guide RNA as a set of GPS coordinates. It tells the Cas9 scissors exactly where in the DNA to make a cut.
And this massive library covered 19,150 different mouse genes.
They essentially targeted the entire genome.
But they didn't just dump the virus in. They carefully controlled the infection rate to hit only about 20 to 30% of the cells. Why that specific percentage?
That math is crucial. By keeping the infection rate low, they ensured that almost every infected cell received only one guide RNA.
So you want to knock out exactly one gene per cell.
Exactly, because if you knock out 5 genes in a single cell and it turns into a supercancer, you have no idea which of the 5 genetic cuts was actually responsible.
It's like giving one unique faulty blueprint to every single runner in a massive marathon.
That's a great way to put it.
So after tagging millions of cells, they injected them into recipient mice. But first, they hit those recipient mice with lethal gamma irradiation.
Yeah, to wipe out their existing bone marrow, creating a blank slate.
So the genetically tweaked stem cells take over completely.
And then the trap is set. You have thousands of different cells circulating in these mice. They all have the hyperactive MYC mastermind driving them, but each cell is missing one different random gene.
And the logic is simple, right? Whichever gene deletion makes the cancer grow the fastest and most aggressively will outcompete all the other cells.
Yep, it will multiply out of control and form the dominant lethal tumor.
Okay, let me make sure I'm fully tracking this massive setup, and we'll do a quick 5-minute recap for you listening before we look at the results. Step 1, we have MYC, a mastermind cancer driver we can't drug without hurting the patient. Step 2, researchers engineer fetal stem cells to overexpress MYC, and then use CRISPR to knock out 19,000 different genes one by one across millions of cells. Step 3, they put those cells into living mice to race against each other. So imagine you are placing a bet on this biological gladiator match. What kind of cellular system, when suddenly broken, do you think would cause a cancer to just explode past the finish line?
To see who won the race, we just look at the timeline.
Right.
The control mice, the ones with the MYC traffic jam, but a harmless fake CRISPR cut, they develop lymphoma in about 140 days.
Here's where it gets really interesting. The mice that received the full 19,000 gene CRISPR library, their average time to develop cancer plummeted to just 74 days.
The gladiator arena worked.
It totally worked. Some of those specific genetic cuts were giving the cancer cells a massive speed boost.
And the researchers actually included a positive control in the race, a known heavy hitter, just to make sure the system worked. That was the P53 gene.
Oh, P53. In the biology world, P53 is famous as the guardian of the genome.
It is the ultimate quality control inspector. It triggers cell suicide if the DNA is damaged. And when they specifically knocked out P53, the mice developed cancer in a terrifying 25 days.
Just pouring absolute gasoline on the fire.
Literally. When they sequenced the fast-growing tumors from the full library mice, P53 was the most common hit, proving the experiment was flawless.
But finding P53 wasn't the goal. I mean, we already know about it, and ironically, P53 is also notoriously difficult to drug.
Exactly. They were digging through the data for the unexpected hits.
And sitting right below P53 in the data were 3 genes popping up over and over. NPRL3, DEPC5, and later they verified NPRL2.
Right.
Now, if you aren't a molecular biologist, that just looks like a randomly generated Wi-Fi password.
It really does. But those 3 genes physically bind together to form a very specific protein complex called GATOR1. Yeah. And when researchers took fresh cells and used CRISPR to knock out just NPRL3 or just DEPDC5, the cancer accelerated almost as fast as deleting the famous P53.
Wow.
It took about 30 to 40 days. Removing any single piece of the GATOR1 complex completely shattered the cell's ability to suppress the tumor.
And finding GATOR1 led to a fascinating discovery regarding a concept called Mutual exclusivity. Can you explain that?
Sure. Normally, a MYC-driven cancer is under immense biological pressure to shut down the cell's suicide pathways. So in about 30% of natural cases, the cancer spontaneously mutates and breaks that p53 guardian gene we talked about.
Right.
But when the researchers analyzed the lymphomas where Gator1 was knocked out, they found something stunning. 100% of them had a perfectly healthy wild-type p53 gene.
Wild-type just being geneticist shorthand for factory settings normal.
Exactly. And to prove those genes were actually functional, they treated the Gator1 mutant cancer cells with a drug called Netlin-3A.
Netlin-3A, what does that do?
It's a chemical that only kills a cell if its p53 system is fully intact. And the drug absolutely decimated the Gator1 mutant cells.
Oh, wow. So it's almost like p53 is the massive steel vault door of a bank. Usually, the cancer has to blow up that main vault door to survive. But Gator1 is an entirely separate secret alarm system on the side windows.
That analogy perfectly captures the mechanism.
So what this mutual exclusivity tells us is that if you clip the wires on the Gator1 alarm system, the cancer is so successful, it grows so fast, it doesn't even need to bother breaking the main p53 vault door. It just bypasses the security entirely.
Spot on. And looking at the broader clinical picture, this mutual exclusivity isn't just a quirk of laboratory mice.
They looked at human data, right?
Yeah, they pulled human data from patients with diffuse large B-cell lymphoma, or DLBCL. In human tumors, mutations in Gator1 components in p53 are also mutually exclusive.
So if a human cancer breaks the Gator1 alarm, it leaves the p53 vault door alone. Okay, time for our second checkpoint to make sure we've got all the pieces on the board. We let the engineered cells race in living mice. The famous p53 gene was the expected winner, but this obscure Gator1 complex was the massive shocker. Knocking out Gator1 made the cancer grow so aggressively it didn't even need to mutate other survival genes. But this leaves a massive, glowing question. Think about this for a second. Why on earth would losing a relatively unknown complex like Gator1 give a cancer cell such an overtowering, unfair advantage?
Right. What is it actually doing inside the cell?
Exactly.
To explain the how, we really have to zoom in from the genetic blueprints down to the actual molecular machinery. We have to introduce the main engine of cell growth, which is mTORC1.
mTORC1. That's it for mechanistic target of rapamycin complex 1.
Yes. mTORC1 is the master control switch for growth and metabolism. When mTORC1 is turned on, the cell goes into construction mode. It builds proteins, manufactures lipids, and grows larger.
And when it is off.
The cell conserves energy, stops growing, and recycles old parts.
So if mTORC1 is the gas pedal for the cell, where does Gator1 fit into the vehicle?
Gator1 is the brake pedal, but it's a highly specialized brake. It functions as an amino acid sensor.
Amino acid sensor.
Yeah, it constantly samples the environment around the cell, specifically looking for 3 vital building blocks: leucine, methionine, and arginine.
Okay, let's use a quick mnemonic to lock those 3 amino acids in. Leucine, methionine, arginine, LMA. Think of it as the leave me alone signal.
I like that. Leave me alone.
Right. If those amino acids are missing, the cell is starving. Gator1 senses the lack of food, yells leave me alone, and slams the brakes on mTORC1. It forces the cell to stop trying to grow when there are no building blocks available.
Exactly. When LMA is absent, Gator1 shuts down mTORC1. But consider what happens if you use CRISPR to genetically delete Gator1.
The sensor is physically gone.
Right. The brake line is cut.
Wait, does that mean a cancer cell missing Gator1 basically hallucinated that it's at an all-you-can-eat buffet forever, even if it is completely starving?
That is exactly what the laboratory data confirmed. Seriously.
Yeah. The researchers took these Gator1 deficient lymphoma cells and placed them in a starvation medium. They physically removed all the amino acids and nutrients.
So a normal cell would detect the starvation and shut down its engine immediately.
But in these mutant cells, the researchers measured a protein marker called phospho S6.
Oh, phospho S6. If mTORC1 is the engine, phospho S6 is basically the dashboard engine light that tells you the engine is revving at high RPMs.
Yes. And even in total lethal starvation, that phospho S6 engine light was glowing bright red.
Wow.
The mTORC1 pathway was locked into absolute metabolic overdrive.
The researchers actually used RNA sequencing to look at what the cell was actually doing with the gas pedal taped to the floor, and the consequences are wild. The cells showed sky-high expression of genes for lipid and cholesterol biosynthesis.
Which makes sense, because to multiply, a cancer cell needs to build billions of new cell walls.
Right, and those walls are constructed entirely out of fats and cholesterol.
They also saw a massive spike in the pentose phosphate pathway. which is essentially the cell's internal sugar refinery used to manufacture the physical backbone of DNA.
Exactly. They even used a chemical marker called OPP to physically measure protein translation in real time. These mutant cells were just furiously churning out new proteins nonstop.
But there's a really crucial detail here that completely changes how we view this vulnerability. You might assume that because the cancer is surviving so well, this hyperactive engine is also producing shields to stop the cell from dying.
But it wasn't.
It wasn't. They checked the mitochondrial function and it was completely unchanged. They measured survival proteins like MCL1, which actively block programmed cell death, and those levels were identical to normal healthy cells.
The cancer wasn't surviving because it built better armor. It was out competing everything else purely because of this massive hyperactive metabolic engine.
All right, final checkpoint on the biology before we get to the real payoff. Gator-1 is the brake pedal that senses the LMA amino acids to tell the cell if it has enough food. Without it, the mTOR-C1 gas pedal is permanently stuck to the floor, pushing the cancer into massive metabolic overdrive. It's building fats, sugars, and proteins without actually changing its death pathways. So consider this scenario. If you have a vehicle with a massive engine, the gas pedal taped down and the brakes completely removed, what is the best way to stop it?
You cut the fuel line entirely.
Yes.
In biological terms, we call this exploiting pathway addiction.
This is absolutely my favorite part of the research. The cancer's greatest strength becomes its fatal flaw. Because the cancer cell is mutated to rely entirely on this hyperactive mTOR-C1 engine to outpace all the other cells, it becomes fundamentally addicted to it.
It literally forgets how to survive any other way.
And this creates a classic therapeutic vulnerability.
Right. The researchers reasoned that if they could shut down mTOR-C1 directly, these fast-growing Gator-1 deficient cells would crash and burn. So they introduced two well-known, already existing mTOR-C1 inhibitor drugs, rapamycin and taurin-1.
And in the petri dish, the results were stunning. Right?
Yeah.
They applied the drugs, and the Gator-1 mutant cells just melted away.
They were incredibly sensitive to the inhibitor.
Meanwhile, the control cancer cells, the ones that still had normal Gator-1 breaks, just shrugged the drug off and kept living.
Exactly.
But, as you pointed out earlier, a plastic dish is a terrible imitation of a living body. How did they actually prove this works in vivo if the cancer cells were engineered with Cas9? Because, well, Cas9 is a bacterial protein. Wouldn't the living mouse's immune system just attack the bacterial protein and clear the tumor naturally, ruining the drug test?
That is a massive experimental hurdle, and their solution was highly elegant. They used RG1 deficient mice.
RG1 deficient?
Yeah, these are laboratory mice genetically engineered to lack mature B and T cells. They have absolutely no adaptive immune system.
Which means they physically cannot recognize or reject the bacterial Cas9 protein.
Exactly. So the researchers injected the aggressive, fast-growing Gator-1 deficient lymphoma cells into these immune-compromised mice. Once the lethal tumors established themselves, they treated the mice with rapamycin for just 5 consecutive days.
5 days of treatment. What was the outcome?
Single-agent rapamycin practically cured the mice harboring the Gator-1 deficient lymphomas.
Just cured them.
The tumors regressed dramatically, and the mice survived long-term. In stark contrast, the control mice who had lymphomas with normal Gator-1, they didn't respond to the rapamycin at all. Their tumors kept growing and they succumbed to the disease within 30 days.
So what does this all mean? We managed to cure a very specific, heavily engineered mouse of a specific lymphoma. How does this translate to human beings sitting in a clinic?
It translates directly to precision oncology. The researchers took this mouse data back to those human DLBCL patient cohorts.
Lymphoma patients.
Right. They isolated patients who naturally had Gator-1 low expression, meaning their tumors were genetically in the bottom 25% for producing those 3 sensor proteins.
And what did they find?
Those specific human patients had significantly poorer survival rates compared to everyone else.
Which means we didn't just find a mechanism, we found a clinical biomarker.
Precisely the point. Gator-1 screening could become a massive tool for oncologists. If a patient comes into the clinic with a MYC-driven lymphoma, you sequence their tumor's DNA.
Right.
If you see that the Gator-1 complex is mutated or expressing at low levels, you instantly know 2 things. 1, they have a highly aggressive, metabolically hyperactive cancer. And 2, because of pathway addiction, they might be incredibly responsive to mTOR inhibitors like rapamycin.
You can hit them with the exact drug their tumor's engine is addicted to. That is just incredible. To synthesize this entire journey for you listening, we started with MYC, a mastermind cancer driver, responsible for 70% of human tumors that we simply cannot drug without harming the patient. So, researchers built a massive living gladiator arena inside mice, using CRISPR to knock out 19,000 different genes to map out MYC's blind spots.
And they discovered that the obscure Gator-1 complex acts as a crucial brake pedal by sensing a few simple amino acids.
Right, the LMA. When cancer breaks that pedal, it goes into unstoppable metabolic overdrive. But that very strength creates a blinding addiction. And by starving that hyperactive engine with an existing drug, an undruggable cancer can be stopped dead in its tracks.
It completely reframes how we look at targeting complex diseases. We don't always have to attack the main undruggable driver directly if we can find the exact supporting mechanism it is leaning on and, well, kick that leg out from under it.
And that leaves us with a fascinating lingering thought. If a cancer cell's most terrifying superpower, this unstoppable hypermetabolic overdrive, is actually driven by the loss of a mundane, everyday nutrient sensor, it makes you wonder, what other simple, hidden switches are secretly controlling the deadliest diseases we face?
It really opens the door to asking what else is hiding in our biology, just waiting for the right genetic screen to uncover it.
It really does. Thank you for joining us on this deep dive into the hidden machinery of cancer. Keep questioning the science around you, keep looking for the hidden back doors, and we'll catch you on the next deep dive.